So it's a tremendous pleasure to be speaking today to you about genetics on the cutting edge. The title of this session is called News from the Genetics Front, and I'm going to give an introduction or primer and leave most of the new findings to the investigators that follow me. So there are two types of genetic alterations in cancer. One is the hereditary changes. These are present in the germline and all tissues in the body. In other words, these kinds of genetic changes can be passed from parents to their offspring. These mutations are almost always retained in the tumor tissues, and usually if the mutation involves a tumor suppressor gene, the second copy of that tumor suppressor gene can be mutated as well or deleted. All genes come in pairs, so for tumor suppressor genes, you generally have to knock out both copies. There are exceptions to that. They're heritable in a Mendelian fashion, so the gene itself can be passed to offspring with a 50-50 chance of being passed to the children of a person who is affected. The germline carriers have an elevated risk of certain kinds of cancers, and the kind of cancer will depend on the specific gene that's mutated. A completely different class of genetic changes are those that are acquired during the disease. These are called somatic. They occur in the body tissue, not generally in the germ cells or sperm and egg cells. These are restricted to the tumor cells, and the mutations have implications for such things as prognosis and what's called now precision or personalized medicine. Some of these changes can help us to define the best therapy that might work in a particular individual's tumor. All tumors are different. Some tumors, such as chronic myeloid leukemia, classically have a single chromosome alteration, the Philadelphia chromosome translocation. But most of the solid tumors have many genetic alterations, and by identifying the breadth of genetic changes in a personalized way, one has a better idea which genes might serve as targets or indicate pathways that might be targets for therapies. So before there was ever molecular genetics, people could look at the genome by looking at the chromosomes. In a normal person's cells, there are 46 chromosomes. These are the chromosomes from one cell of a mesothelioma. You can see that there are some chromosomes that are missing. I hope I don't move the slides when I do this. But you can see that there's a missing 2, there's a missing 22. So there are less than 46 chromosomes. But in addition, all of the chromosomes that have arrows indicate that there's something structurally the matter with those chromosomes. Usually it's a deletion of a part of a chromosome. And we now know that the genes that are responsible for mesothelioma seem to have been right before our eyes when we looked at cytogenetics. Both copies of these chromosome 9s have a part of their chromosome that's abnormal, and we now know that there's a gene that encodes P16 and ARF. These are two tumor suppressor genes that are involved in the two of the most important pathways in cancer, the retinoblastoma and P53 pathways, and they're usually homozygously deleted. There's another gene on chromosome 22, another tumor suppressor gene. One copy is mutated, and the second copy, you can see the second copy of chromosome 22 is lost, so that second copy of NF2 is lost. So again, you have both copies of a gene altered. And way back in the 1990s, we had identified that there was a region where there were subtle deletions in chromosome 3 in at least 60% of mesotheliomas, and we narrowed this down to a region that Mark Ladani's group subsequently found, and we confirmed that there are somatic mutations of a gene called BAP1, which will be the subject of much of what we talk about today. So There are also somatic mutations. Those were somatic mutations I was referring to in mesothelioma in that previous slide. These are also somatic mutations in BAP1 in uveal melanoma, which is a kind of eye cancer. You can see some of these eye cancers, this one is in the cornea, in the iris rather. This one is in the ciliary body, and this is a massive tumor that Dr. Bill Harbour will speak about in a moment. He had a front page article about several months ago in the New York Times in terms of personalized medicine. In other words, it's not the size of this tumor. This is a massive tumor. This doesn't necessarily mean it's a bad prognosis. This one could actually have a bad prognosis if it has the BAP1 mutation, because what Dr. Harbour found is that if you have a mutation of BAP1 somatically, it means that there's a great likelihood that it's going to metastasize, that tumor will move to the liver and will kill that patient. So it's an incredibly important marker of a very bad prognostic indication in a particular patient's tumor. And what he had seen, this is so recurrent that almost 85% of patients who have metastasizing tumors have this mutation. Now, for those of you who've never seen a pedigree, I'll now move to hereditary mutations. This is a high-risk cancer family. It's a classical BRCA1 family. Many of you have heard of BRCA1. It's a mutation that occurs in the germline and be passed from one generation to the next These 1, 2, and 3 indicate the generations. Males are indicated by the boxes. Circles indicate the females. All of these females are getting either breast cancer or ovarian cancer, and they all have a mutation of a tumor suppressor gene known as BRCA1. There are many examples of cancer syndromes so BAP1 isn't the only one. There are at least 25 that I could identify here where you can have a mutation of a gene that might predispose to multiple tumor types. So it's not unprecedented for a mutation to lead to a family having several different kinds of cancers. So you can see here, if you have a mutation of either BRCA1 or a related gene called BRCA2. You can have breast cancer in the family, ovarian cancer. If you have BRCA2, you can also have prostate cancer, pancreatic cancer, and even melanoma. If you have a mutation of the P53 gene, another tumor suppressor gene, this causes Li-Fraumeni syndrome. And you can have breast cancers, carcinomas, sarcomas, brain cancers, adrenocortical cancers, and in fact there's even been an example of a patient who had a P53 mutation in the germline who developed mesothelioma. So I became involved as a result of Janet Rowley suggesting to Dr. Michele Carboni that he contact myself and also investigators at the University of Chicago, Nancy Cox, who was looking at ways to identify genes using a technique called linkage analysis. So this was part of a large program project with the study rationale listed here being that generally only a small fraction of all asbestos-exposed individuals, even individuals who work in asbestos mines, probably only about 5 to 8% of individuals that will develop mesothelioma. Why is that? Why don't you have 100% of them developing mesothelioma? On the other hand, you get clustering of mesothelioma in some families, and therefore we think that some families harbor a genetic predisposing factor, and this study led by Dr. Carboni eventually did find that that was the case. So we had detailed pedigrees that Haining Yang and others developed, and we had blood from these individuals, both the affected individuals who developed mesothelioma and other members of the family. And we also had just a few cases where we had solid tumors from those same families. So the blood was used for the hereditary studies, the germline studies, and the tumor was to look for somatic mutations. Sometimes if you identify a somatic mutation, it can tell you what the hereditary change might be. So both of those studies were important in leading to the discovery of this gene. Neither one of these two families, one in Louisiana and one in Wisconsin, neither one of these families had exposure to high levels of asbestos or erionite. There were no occupational exposures in the families, although there were trace amounts of asbestos found in their homes. So these are the two families. All of the orange, just to make it simplified, I have all of the orange indicating mesothelioma throughout all the slides I'll show. So you can see that this disease is being passed from generation to generation, from these parents here to their offspring and to the third generation. Some of these individuals have breast cancer as well as mesothelioma, for example. In this family, you have two that are highlighted in green. These are individuals who had those eye tumors, the uveal melanomas, including one patient who had both mesothelioma and uveal melanoma. And that's going to be something that is repeated over and over again, just as it is in some other kinds of cancer syndromes. In fact, there was a paper in Science just this past week where one woman, it wasn't a BAP1 mutation, it was actually something that's involved in telomerase, which Dr. Carboni and I studied maybe 7 or 8 years ago, where a mutation in this gene actually led to one woman having 6 different cancers. So Dr. John Min-Pei and my group had, about 2 years before we published this work, come to the conclusion that he had seen that BAP1 was actually the gene. What we show here is just a segment of chromosome 3p21. It's within a much longer region within this chromosome. And what he identified in these two tumors, one from Wisconsin and one from Louisiana, that there were mutations, both of which targeted either the loss of BAP1 in this case, both copies of BAP1 were lost, or in this case, there was a mutation in a region that regulates the expression of the gene. It's called the promoter part of the gene. So in both cases, he thought this BAP1 was the gene. We weren't entirely convinced because Nancy Cox's group was doing linkage studies, and on the family W, it strongly implicated a small region in a completely different chromosome, chromosome 6. However, later linkage at that chromosome vanished when a presumed unaffected person developed cancer, I think in their 80s. So linkage investigators have to make certain assumptions, and one of the assumptions is that, well, if a person doesn't have a cancer by that age, probably they're not affected. Once they looked at that person's genotype, it's completely different than the other members of the family, so we were really depressed about that. Dr. Masaki, or Mr. Masaki at the time, was working on his PhD and was sequencing the entire chromosome 6 region and found no mutations, and of course he wouldn't because that wasn't really the site. So we were really fascinated. One afternoon, Mitchell Chung and my group came to me and said, look, here's this paper. And I had always been fascinated that mesothelioma and melanomas have a number of similarities. For example, they can both have alterations of P16. But the key point in Dr. Harber's paper for us was right here. One of the tumors harbored a frameshift mutation that can totally destroy the protein product, and it was germline in origin. So this was really exciting for us, and we immediately started to sequence. And Mitchell found, I won't go into the details, but this is a way that scientists look at genes with this kind of fluorescent tag. You see greens and reds and blues and so forth. And what he found is that in both families, there was a mutation that changed one nucleotide. Now, you remember there are 3 billion nucleotides in the genome, so this would be like finding one uncapitalized letter in an encyclopedia. He found that mutation, And it turned out that mutation makes this gene produce a protein that is abnormal. It can't get where it's supposed to be in the nucleus. In the other family, he found that there was a stop codon, which immediately, in the middle of the protein, truncates it and makes it no longer be able to produce a full protein. So this was really exciting to us. And then Dr. Carboni's group, Masaki Nasu, who was sequencing earlier chromosome 6, now started to sequence sporadic tumors that were obtained from Dr. Pass and Mary Hesdorfer. And what they found were germline mutations of BAP1 in 2 patients out of 26. These are sporadic, and they're finding these mutations in their blood, in their germline. So it means that maybe some patients who don't have a whole family with mesothelioma can have a germline mutation. Sometimes it might be just one that occurs de novo. It wasn't in the whole family. It just started to happen. This kind of thing happens all the time in our bodies. We're always sustaining mutations. And if it's in the germline, that could be passed to your children. Both cases with these BAP1 mutations had frame shifts. That means that there's a change in the sequence order, so you no longer make a functional protein. And in fact, it truncated the protein, so it was no longer able to go where it's supposed to. And importantly, both sporadic mesothelioma patients had, who had this germline mutation also previously had uveal melanomas. Now, the chance of having those 2 tumors together is extremely rare. The statistician that worked with us said it was something like 1 in a trillion that you could have a cancer that occurs only 3,000 patients a year for mesothelioma and only 1,500 patients a year for uveal melanoma. To have both of those, the odds are incredible. Now we know in the literature there are now at least 6 cases like this, including 3 that we haven't published yet. At the same time that our paper was published, Dr. Weissner and DeSpiker reported in the same journal, unbeknownst to us, they didn't know about our work either, 2 families that had BAP1 mutations. But unlike ours, they found all of these melanocytic lesions throughout their bodies. They look like this. Probably some of you think you have one like that on your arm. I thought I did, but I'm a hypochondriac. But, you know, they don't look like much. They're a little raised, dome-shaped. They're not black. They're slightly pinkish, reddish. When you look at them by pathologist view, then you start to see they don't look right. Some of these nuclei, the dark staining things, are big. Some of them are small. Sometimes the cytoplasm is incredible. And so this reveals a proliferation of these large epithelioid melanocytes. So interestingly, if you look at this family, there were also, so all of these black and gray indicate, these symbols here indicate that they had either these papillary melanocytic tumors that are all over the place, and now Dr. Carboni's group has found that our 2 families from Wisconsin and Louisiana also have some of these. Maybe not as many as these families, but they do have some, and he'll talk about that. And you see the 2 that have red symbols. These 2 had uveal melanomas. So once again, uveal melanoma is coming up. And more strikingly, Dr. Spiker wrote me an email and said, interested in your article, by the way, did any of your patients have these lesions? Because one of our patients now has turned out to have mesothelioma. And he went on to report another family that has 4 or 5 mesotheliomas in the family, and only one of those patients had these lesions. So we don't know why. Why do some people manifest this syndrome as mesothelioma? Why do some others manifest it as a melanocytic lesion? Why do some that have a melanocytic lesion change to have part of that lesion become malignant? Is it because of environmental exposures to sun, to asbestos? This is all the area that we're all excited about working on right now. Most of the things we do in life, everything from the intellect of your child, has a component that's genetic and has a component that's environmental. My guess is the same will be the case here. This is a new family that we've studied, and I wanted to point it out because we actually have 4 different members of this family that have 2 different cancers. The odds of that are very, very unusual, but Jill O'Hara will talk about some of the evidence that she has that it may not be as rare as we think. And again, you see the uveal melanomas coming up in a patient who also had mesothelioma. So I'll turn now to the speakers about the most recent exciting results. Our overall goal for this session will be how do genes play a role in mesothelioma, particularly with regard to a new hereditary cancer susceptibility syndrome that's being called the BAP1 cancer syndrome, which predisposes to malignant mesothelioma, which is the MM, uveal melanoma, cutaneous melanoma, atypical melanocytic tumors such as the ones I was showing you earlier, and potentially other cancers such as kidney cancer, perhaps breast cancer, perhaps meningiomas. And what are the future implications of genetics, markers, and inflammation to this field? So I believe the first speaker is Dr. Carboni. Is this list correct?
I think we might go a little bit out of order. I think we might call Dr. Passa.
Okay.
And then I just want to make just a quick announcement. If you have questions for this panel, if you could write them down on blue index cards, because we're taking questions from Facebook and the audience, and we want to make sure that we sort of streamline the questions. Thank you.
So some of our speakers are incredibly busy physicians. You heard earlier, for those who were in the new patient session, that many of these surgeons are on call at any time for their patients, Sundays, whatever. And so for them to be here is an honor, and to have them speak, and some of them do have to get back. So our first speaker is a surgeon, Dr. Harvey Pass at NYU. Dr. Pass has nearly 400 papers on the area of mesothelioma. You remember Mary Hesdorfer said that it's important that if you consider a surgeon, that you consider surgeons who really are in this field. This is not a game that you do occasional surgeries. You have to do a lot of these surgeries to do them right. Dr. Pass is the Stephen Banner Professor of Thoracic Oncology, the Vice Chairman of Research in the Department of Cardiothoracic Surgery, and Director of the Division of Thoracic Surgery and Thoracic Oncology at NYU's Langone Medical Center and School of Medicine. It's my pleasure to introduce Dr. Pass.
Thanks, Joe. For me, it is a game. The game is to find a cure. So I'm really happy to be here at MARF again, or the Meso Foundation. Obviously, I've worked with everybody, and obviously I've worked with the BAP1 people. This is not going to be about BAP1. I don't care whether you have a BAP1 mutation or not. I mean, I'm interested in just finding mesothelioma early. So that's what we're going to concentrate on with my lecture. And I have to disclose now who I work with. So I work with these people, and I get grants from these people, and I have some patents. So what's the magnitude of the situation? The magnitude is that 27.5 million people are going to be exposed to mesothelioma, or have been. We think that there's going to be a peak in the mesothelioma incidence in the next 35 years. But the financial burden of this, not only on the families, but on the countries, is extraordinary, as you can see. And in the United States, it's a select few of 3,000 about. There's no real registry, but we think that's what it is. And we got to find it, but it's somewhere between 10 and 40 years. So that's a long time to be surveillancing somebody who's exposed. Now, why would we want to do this? Well, this is my own personal experience in operating on mesothelioma. The big line that's over on the right are the patients that have a median survival of 48 months. Not everybody dies of mesothelioma, okay? So you need to be able to move these curves here that are the late-stage patients up here, so that then the stuff that Mary was talking to you about, not only with surgery, can get the line up here. And that's what we want to do by trying to find things earlier, because these are the early stage patients that get operated on, and they do the best. So what makes for a good marker to try and find disease early? And what do you have to do to make sure it is a robust marker? And how do you have patients to be able to test this in? Well, good markers have to be sensitive. You want to have a marker that you can measure at such a low level that that low level is going to say it's a meso or not, or a patient is at high risk or something's going on. It also has to be specific. I'm not interested in markers that can find patients with inflammation from asbestos and also show that a patient has a mesothelioma. No, it has to distinguish patients who have asbestos but don't have meso, and patients that have meso, and also patients who have lung cancer and patients who have ovarian cancer, because we're interested in specifically finding meso. But you're not going to screen everybody in the world for mesothelioma. So at least at the beginning, you have to screen high-risk individuals, grossly people in these families that you've heard about that may be familial mesotheliomas, certainly they fit, and also people who have had an exposure to asbestos and a documented exposure. So how are you going to do this? So remember, you don't have a CT on everybody, so you have these people who are coming to see you. Now, which one of these patients are you going to be most concerned about? They both have asbestos exposure. This person does not have any fluid in his chest. This person does. But these patients come off the street. You haven't done the CT yet, and they have no symptoms. Because by definition, screening means you're screening asymptomatic patients. So you're going to miss this patient who has the effusion. And that effusion could be mesothelioma if you don't have some way of knowing when the patient visits you. I personally believe you ought to have a blood test. You ought to have a blood test that tells you something's going on, that then points you to get the CT scan, that then shows you there's a difference between this guy, and you better do something about this. So essentially, the bottom line here is that most of the patients will present with some sort of effusion. You're looking to find a marker that says a patient has an effusion, and you're looking to find a marker that says the effusion is a meso. But how do you even determine who had a significant history of asbestos exposure? You know, you speak to people who work in the industry. Yeah, I worked 10 years, but I really didn't go to the houses and take out stuff. Yeah, I've been working 40 years. I never wore a mask. I got the dust all the time. Those are different risk profiles of patients who could develop mesothelioma. So wouldn't it be nice, at least, if you had some sort of marker that said, oh, that guy got exposed with asbestos? Then you know who to screen for meso. Well, it has to do with all these pathways that asbestos does, that maybe it will elicit something in the blood that you could measure from all this, that may be just an inflammatory sort of protein. Well, from Dr. Carbone's lab, and you'll hear from Heining Yang and from Michele about this, they're interested in a marker called HMGB1. And if you look at the levels of HMGB1 in patients who are non-smokers and not exposed to asbestos, patients who are smokers but not exposed to asbestos, and then patients who are asbestos exposed but don't have meso, you can see there's a difference between normal so-called people and patients who have been exposed to asbestos with this marker. There's a marker in the blood. But that marker persists if you have mesothelioma. So it's not going to distinguish between a patient who has asbestos injury versus mesothelioma. But it's pretty good maybe as a first line. This needs to be validated. Hopefully it'll be validated in a big NIH grant. So how about markers that are going to distinguish asbestos-exposed patients from meso patients? Well, there is a litany of this, and I'll talk about some of these. The gold standard, the granddaddy, is the SMRP, the mesomark. And that is a true marker. It's not the best marker in the world, but it is a marker that you should ask your physicians about because you can get it measured at a reference lab, although the FDA doesn't measure it. It can be measured at a reference lab. And this is a study that we did that was a blinded study in which we had 2 places look at sera from patients who did or did not have mesothelioma. And what you found was that if the marker was no good, it would fall right on this 50% line. Well, SMRP at both places was able to distinguish asbestos from meso very well, and also in stage 1 and stage 2 cohorts, early patients with mesothelioma was able to distinguish not quite as good, but still there's a signal. So SMRP is good for a baseline, and then you get treated and you see what happens. Here's another one from our lab that's called osteopontin. And we published this a long time ago. Well, 2005 is a long time ago. And this marker looked great. It was an area under the curve. It was 0.85 for SMRP. This was 0.9. And these are asbestos people, and these are mesos. You can see the difference in the levels, and everybody's excited because this is stage 1, and there's a difference. Big hullabaloo. Problem is, this molecule falls apart in serum over time. So it's not very good if it's left around standing, not like SMRP, which is good for a long time. So what happened is that other people tried to duplicate this and they couldn't find it. But what they found, though, was if they looked in plasma, it did replicate. Now, what's the difference between plasma and serum? Plasma doesn't clot. Serum clots. So you can imagine when you make a clot in a tube and you're trying to measure a biomarker, all hell breaks loose with a marker that maybe has what's called a cleavage site, which osteopontin does. So when you start clotting, the cleavage site breaks and you lose the marker or you get fragments. So plasma is the answer to your cures for looking at biomarkers, in my opinion. So all of our stuff ever since has been done in plasma. Now, you know, when I was disappointed about this first marker, I said it's time to go back to the sweatshop and find some more markers. So we did that. Now, how do we do that? Well, this is the way we do it. We look at the genes that are different between tumor and normal. And how can we do that? Well, I can't show you. Yes, I can. So here's a meso specimen. This is the lung, and this is the peritoneum, which is the lining from the inside of the chest, inside of the belly that comes out with it. This is not affected by mesothelioma. This is. So if you take a piece of this that's normal, but is the mother tissue that makes mesotheliomas, mesothelium, then you're comparing apples to apples with regard to the normal tissue and the real tumor. So we did that and did a whole bunch of genes that we looked at. The ones in yellow are some of the interesting ones. This is our old friend osteopontin, so that validated again. But there was one that I'd never heard of, which is down here, which was a strange thing called EFEMP1 or fibulin. I never heard of it, never had any ideas. But there was some stuff in the literature about it being elevated in glioblastoma. Well, it turns out that glioblastoma, brain tumors, have a lot in common with mesotheliomas. Okay, so cut to the chase. This is what we published on this marker. This is plasma from patient's blood comparing to other patients' blood. So we're comparing mesothelioma levels of this marker to all the rest of these patients. And here you are with patients who have an asbestos exposure. So their levels, and that's mesothelioma. And then you have the rest of these. And notice, these are patients that have a lot of effusions. Remember I talked about finding effusions before, about screening and that sort of stuff, and specificity? Well, these are all sorts of cancers that make fluid, but the levels of the fibulin are not nearly as high as with mesothelioma. And remember, this is not measuring the fluid, this is measuring in the blood. And so the sensitivity and specificity was great, and we can show that you can find it in early disease, stage 1 or stage 2. And lo and behold, if you operate, even if I operate on the patient, you can show that the levels will go down, which is a good thing. And then, unfortunately, if the patients recur, the levels go up. So maybe this is a marker that you can follow patients to see how they're doing also. It doesn't mean anything unless somebody else can do the data and say that it works. And my good friend sitting in the audience, Dr. D. Perot, partnered with me, and he sent us his samples of mesothelioma and asbestos, and I had no idea which was which. And all I did was measure fibulin from them, and then I sent our data back to him, and he told me whether we failed or not. Well, fortunately, we didn't fail, and it replicated pretty well. It's never 100%, but this is a pretty good single marker replication for mesothelioma versus asbestos from the University of Toronto, so we were encouraged by that. You can also measure this in effusions in the fluid itself from the patients. And the levels are extraordinarily high compared to other patients, meso, other patients, and you can even use it to discriminate from effusions from other people or from the plasma. So you see, if you look at effusions from other types of effusions, there's a big difference between meso and other. If you look at benign effusions, things that happen not from cancer, but you look at the level of fibulin there, it's very low compared to meso. And if you look at other malignant effusions, lung cancer, ovarian cancer, breast cancer, lymphoma, that's the level of fibulin. There it is in mesothelioma. So for what it's worth, it looks like fibulin, looks like a pretty good marker. And also, if you look in the fluid, you can also see that certain patients who have low levels of fibulin in the fluid do very well, while patients with high levels don't do very well. So maybe we can discriminate who we should be aggressive with, who should not get surgery, who we know is not going to have a good result with surgery. So, you know, now, what are other ways that you can look at markers in mesothelioma? Well, you need to know who's doing the research in industry, because industry has the toys that we don't have in academics sometimes. You can actually measure proteins in the blood by using DNA-like moieties, because these DNA, these nucleic acids, actually bind to proteins. So if you manufacture a whole bunch of nucleic acids that are specific for binding certain proteins, you can put the whole gamish together, you can elute off the proteins eventually, and then read the levels of the nucleic acids, which is specific for a given protein. Just like a gene array, just like looking at hundreds of thousands of genes. Just that you're looking at proteins. We did the same sort of analysis that we did with fibulin. We looked at patients who had mesos, and we looked at patients who didn't, and we studied and then validated, and then we had other patients that we studied and validated who didn't have mesothelioma. This is a pretty good marker profile also using this platform. What this means is that if you took a certain number of these somomers, which is the way that you measure this with these nucleic acids, the discrimination is excellent, just about as good as fibulin. And it was both in the blinded verification set and also in early stage disease that you could distinguish meso. That wasn't good enough. We did another validation with a separate set of specimens from Sinai and from Libby and found the same verification. So this is another marker, the marker panel, that involves all these put together. Some of them are nonspecific. Some of them have to do with what meso is doing to the normal tissue. A lot of inflammatory cytokines, just like Dr. Carbone's group has shown with HMGB1. So this may come commercial in about 6 to 12 months. So I've shown you what's out there at this point. Obviously, there are other markers that I don't know about that my colleagues are doing that may be just as good, that may be published. But I think we're on the road to finding markers to find this disease earlier. We're on the road to finding markers that will help us tell us how the disease is doing. And the key thing, though, is everybody has to work together and establish a consortium of centers, which we are doing, trying with a SPORE as well as with all the people we work with, to do this together to validate it. And then get more research funding to be able to start prospective screening trials with these markers to make sure they work. And then finally to design the proper protocols to be able to make sure that these validate. Couldn't do it without my lab team, specifically Chandra, who does all the protein work in my lab, as well as all my other people, PhDs and none such. Thanks very much. So do we want to do questions now? I mean, I'm going to be here until 12.
Does anyone have any questions for Dr. Ralph that they want to submit?
I actually had one question. Does SOMAMERS work for serum, or is it, again, plasma?
SOMAMERS works for serum. It will actually work. It will work for plasma also. We've done both.
Very exciting. Now our next speaker is Dr. William Harbour. He was at the University of Washington in St. Louis for 16 years, and just recently has moved to Miami. I guess he had been in Miami in years past for part of his training. He's currently, I don't see him. Dr. Harbour? Maybe we'll move on to someone else. Mary? I don't see him. Okay. So Dr. Harbour is the Vice Chairman for Translational Research and Director of Ocular Oncology Service at the Bascom Palmer Eye Institute at the University of Miami, where he's now directing a multidisciplinary team of physicians and scientists studying the genetics and genomics of the major eye tumors. It's a pleasure to have Dr. Harbour speak to us today.
Well, it's a great pleasure for me to be here, and I want to thank Dr. Testa, Dr. Caborni, and the other meeting organizers for having me. And you probably didn't think when you came here you were going to hear from an eye doctor. So first I want to after showing you my disclosure, explain to you why you would have an eye doctor here. And you've already gotten a hint of this from Dr. Testa. This is a family that came to me after I recently moved to Miami to join the Bascom Palmer Eye Institute. This family is from Louisiana, so I suspect that this contains many of the same members of Dr. Testa and Dr. Carboni's family. But the interesting thing here, as you've already heard, is that uveal melanoma and mesothelioma are really the dominant cancer types within this family. So I'll come back to that later, but first I just want to give you a little background on uveal melanoma or ocular melanoma. The uvea is a strange word that it represents a tissue layer within the eye between the white coat on the outside of the eye, the sclera, and the retina on the inside, which is the visual part of the eye. This is a very vascular tissue that contains a lot of melanocytes. The part of the uveal tract that you're all familiar with is the iris, the colored part of your eye, and you can get melanomas here. You can also get melanomas in the tissue just behind the iris, which is called the ciliary body, and then in the choroid. So sometimes you will hear these melanomas referred to as choroidal melanoma or ciliary body melanoma, but they're all essentially uveal melanomas. The vast majority of patients, we can save their eye these days. Up until a few decades ago, most of these patients had their eye removed. But we can now treat about 90% of them with sophisticated forms of radiation treatment, where we implant devices on the surface of the eye. We localize them with ultrasound, as we see here. And we have about a 98% success rate at saving the eye in these patients. Unfortunately, and just a little bit more background on uveal melanoma, it's about 5% of all uveal melanomas, mostly in Caucasian patients, and generally is thought of as a non-familial cancer. So we'll come back to that in a minute. Despite the successful treatment that I just mentioned, up to half of these patients, if they live long enough, they don't die from something else, will develop spread of their uveal melanoma. And most of the time it goes to the liver, where it's incurable. So we and others have spent a lot of time and effort trying to understand the genetics, the molecular underpinnings of this cancer. And the first thing we found almost a decade ago now is that there are really two flavors of uveal melanoma. Even though they look the same when we're examining the patient, there are really two different forms at a molecular basis. And we now call these forms Class 1 and Class 2. The Class 1 melanomas have a very low metastatic risk, as seen here. The Class 2 tumors have a very high metastatic risk, as seen here. And importantly, the Class 2 tumors are associated with loss of chromosome 3. And it's been known for many decades that loss of chromosome 3 was not a good prognostic factor in uveal melanoma, but nobody really understood why. A couple of years ago, I guess almost 3 years ago now, we had looked every way we knew how on chromosome 3, but there are thousands of genes on chromosome 3, and as Dr. Testa said, you're looking for one misspelled word in the dictionary or the encyclopedia, and it was very hard to do that until recently, when a very sophisticated new technology became available, which all of us in cancer research are now using. It's a way to very rapidly sequence or read the entire genome very cheaply and easily compared to previous methods that we had. And by doing that, we discovered that there was one and only one gene on chromosome 3 that was damaged, and it was only damaged in the Class 2 metastasizing tumors. And of course, this was BAP1, which you've already heard about today. What we initially saw was that the mutations mostly damaged this part of the gene that has to do with its activity as an enzyme. Interestingly, when you look at the certain type of mutation that just specifically damages one part of the gene without destroying it, you can get further insights into what is it about the gene that's so important. And again, most of the mutations cluster in this area that has to do with the activity of the enzyme, but there's also this other cluster of mutations down here, which I'll mention later. And as was mentioned, we were very surprised to find that one of these patients had a germline mutation. In other words, we found the mutation not only in the cancer, but in their peripheral blood, which means it's in all the cells in their body. And when I was in training, we were taught that uveal melanoma is never familial, it's never hereditary. So we were very surprised. We went back and rechecked this, and sure enough, it was definitely the case. Unfortunately, this patient was not available for further questioning and further evaluation, and so we turned to some other families that we knew had a strong hereditary apparent hereditary predisposition, that we had collected blood from the past but really didn't know what to do with it. So when we went back and looked at one of these families, this is my patient right here who had uveal melanoma. They had a sister with uveal melanoma, a brother with cutaneous melanoma, and uveal and cutaneous melanoma on another branch of the family. And indeed, they all had BAP1 germline mutations. In this particular family, gastric cancer was a prominent feature. However, we were not able to actually confirm that those patients had BAP1 mutations because they were already expired. This is the family I just showed you before. where, again, uveal melanoma and mesothelioma are the predominant features, but you do again see gastric cancer, renal cell carcinoma, and a number of other cancers. This is a summary of Dr. Testa and Carboni's work, Dr. Wiesner's work, and a number of other papers. We see a paper almost once a month now of a new family or a new association between different cancers for this BAP1 familial cancer syndrome. This is an example of a patient who walked into my office a couple of weeks ago. He was only 17 years old, and he had a large tumor. Unfortunately, we had to remove his eye, and it's very unusual for a 17-year-old to get a uveal melanoma. And so I started thinking, you know, when you see young patients with cancer, you start thinking maybe there's a hereditary component. And I said, do you happen to have any moles on your skin? And he said, well, yes, I've had quite a few taken off. And I said, can you show me some that are still there? And here you go, this sort of typical BAP1 associated melanocytic nevus or mole that looks different from your typical mole that most of us have. So there's a growing list of cancers associated with this syndrome. The ones at the top of the list, again, are mesothelioma and uveal melanoma. There are a number of other cancers that vary in the degree of scientific rigor with which they have been associated with the syndrome. Now, why is it that so many of us have been studying these different cancers for so many years and never saw this association before between uveal melanoma and mesothelioma, or in uveal melanoma even seeing familial cancer? And I think at least one answer to that is you need to have large families with lots of offspring before you will see the occurrence, because the chance of getting any one cancer is not that high. The chance of getting some cancer is pretty high. But if you're only looking at one type of cancer, you may not see the pattern in a particular family unless you have a pretty large family. Here, this family here had 8 siblings. If they had only had 2 siblings, 2 children, we may not have seen the pattern at all. So for any individual cancer, what we call the penetrance, the chance of it penetrating to the next generation is relatively low. But when you look at the overall cancer burden, it's clearly much higher than you would expect. So I'm going to quickly go through some of the research that we and others are doing, trying to figure out what in the world is this BAP1 gene doing in the protein that it produces that is so strongly associated with cancer. And this is really just a summary slide that says it does a lot of stuff and we don't really understand it yet. The part that was missing from this really excellent review is the association of BAP1 with the control of stem cells. And stem cells, as you've heard, I'm sure, are cells in our body that are not fully committed to become one type of cell or another. We're born with many stem cells in our body, and as we grow and mature, the number of stem cells decreases. As our arm develops, for example, we eventually all the cells in the arm become arm cells, and they cease to become stem cells. However, in cancer, one of the things we're starting to understand is that these cells that were once committed to being a skin cell or a brain cell, they start to drift back to this less differentiated or less committed state where they can start to do mischievous things. And we think that at least in some small way that's what's happening with BAP1 loss in uveal melanoma. We think that BAP1 is somehow controlling the differentiated state of uveal melanocytes. And when you lose BAP1, you start to evolve more towards a stem-like state. This is, for example, these are class 1 melanoma cells right out of a patient where we have knocked out the BAP1, and they start to ball up into these epithelioid cells. And we know that epithelioid morphology is strongly associated with the class 2 gene expression profile and with BAP1 loss, and you've already heard that epithelioid morphology is associated with BAP1 mutation in mesothelioma, and it's also associated with BAP1 loss in cutaneous melanoma. So there's something about this epithelioid morphology that's strongly associated with BAP1. We're not sure what it means, but we think that at a minimum, what it means is that these cells no longer look like melanocytes. They're not sort of star-shaped, differentiated cells anymore. They look more like stem cells. And this is a very busy slide that I'm not going to go through, except to point out that we've done many types of experiments to understand what happens to uveal melanoma cells when you lose BAP1, and they all point to this regulating a stem cell type change in the cells that allow them to drift away from where they're supposed to be and to start doing things they're not supposed to do, like spread to the liver and start growing. We've identified a potential therapeutic target for cells that have lost BAP1. These are called histone deacetylase inhibitors, or HDAC inhibitors. And some of these were mentioned earlier today. SAHA is a HDAC inhibitor. There are even drugs like valproic acid, which have been around for years, that are used for seizures that actually have HDAC inhibitory activity. And what we've found is that when you lose BAP1, you get an increase in something called ubiquitination of histones, which we think might be key to what's going wrong when you lose BAP1. And we showed that if you treat the cells with an HDAC inhibitor, you can reverse this biochemical defect. And that's associated with a shift from these more stem-like cells back to a differentiated morphology in these melanoma cells, and a shift from class 2 back to class 1 in their expression profile, and a reestablishment of a differentiated gene expression profile. And finally, we were able to show in an animal model that valproic acid can blunt the growth of uveal melanoma tumors in the animal. So just want to shift gears here for a second and go back to what I was mentioning earlier, that many of the mutations that affect BAP1 are damaging the activity of the enzyme. But there's this other cluster down here of mutations, both in missense mutations in tumors and in germline mutations, that are not really, they're not directly affecting this catalytic activity. There are a number of other activities down at this end of the gene. One is where BRCA1 binds, and another is this nuclear localizing signal that Dr. Testa mentioned earlier that's required to get the protein into the nucleus. But Dr. Frank Rauscher at the Wistar Institute, who we have been working with to understand some of the mutations that we find in patients, has come up with a really fascinating discovery that that this end, what we call the C-terminus of the protein, is actually required for the catalytic activity at the other end of the protein through a folding of the protein from one end to the other. And when you it has to do with where the active site is located and accessed for the ubiquitin here, which I'm going to skip over. But the bottom line is that these tumor-associated mutations are clustering right in this area that allows this folding of the protein. When you can't get the folding of the protein, then BAP1 no longer binds this important partner called ASXL. There's an ASXL 1, 2, and 3. And when that's blocked, then you don't get the enzyme activity that you need. So he's working on a very interesting strategy for therapy, which is to somehow stabilize this folding of the protein. So this is an interesting area where even though we made these inroads with uveal melanoma, the findings in this basic research may well apply to mesothelioma and other cancers as well. So just summarizing, we've now learned a lot about uveal melanoma, and that this BAP1 mutation seems to be strongly associated with this switch to a metastasizing phenotype. What I didn't talk about today is we more recently discovered a mutation which is mutually exclusive with BAP1 that seems to be associated with a better prognosis. We published this a month or two ago in Nature Genetics. Now, the interesting thing is that whether you develop a uveal melanoma from a germline mutation in BAP1 or whether it's acquired somatically, you still have to have an initiating mutation. The BAP1 mutation does not initiate the cancer. And that's true also in cutaneous melanoma. You need an initiating mutation in something like BRAF. I'm not sure if that's also the case in mesothelioma or not. But there is a certain sequence of genetic events which is necessary for uveal melanoma. So, in closing, I hope I can convince you that even though I'm not studying mesothelioma directly, that the research that we do may have some insights into mesothelioma and vice versa. And in fact, what we're really starting to understand as cancer researchers is that we're thinking less about grouping cancers in terms of their anatomic location and more about their similarities biologically and genetically. And in other words, instead of thinking as all lung cancers is the same, or all breast cancers as the same, or all brain cancers as the same, we really are thinking, well, there are some brain cancers and some lung cancers and some other kinds of cancers that are very similar to each other, and it doesn't really matter so much where they're anatomically located as much as the biological properties that they have in common, because that's going to be what drives our development of new therapies. So in conclusion, uveal melanoma and mesothelioma are the two defining cancers of this new BAP1 cancer syndrome. As far as I know, there was no known association between them before this exciting work came out from many groups almost simultaneously a few years ago. I think one reason that it was not noticed before is that the chance of getting any particular cancer when you have this germline mutation is not high. So you need to see a lot of family members before the pattern comes out. BAP1 mutations seem to be associated very strongly with metastasis, but because of this germline association, it clearly can be involved with progression of the primary cancer as well. And then BAP1, the role of BAP1 in all of these different cancers is going to differ somewhat depending on the cancer type and so forth. But I think what we learn from one cancer is going to be very important for informing our understanding of the other cancers. And I just want to acknowledge the people that have worked with me on this project and our sources of funding. Thank you.
So in case we don't have time to ask questions towards his end, let's do that now. I have one question. You showed one family, no mesotheliomas, but it was interesting. There were, I think, 4 or 5 gastric cancers. Was that an Asian family, perhaps, given the incidence of gastric cancer in Asians being higher?
Yeah, it's a great question. No, it was a typical Midwest Caucasian family. But again, I hasten to add that we were not able to confirm that those patients had a germline BAP1 mutation, that they had already expired.
Other questions?
Thank you.
All right, thank you very much. So is there any other speaker that has more of an urgent need to catch a plane, or can I just go in the order that's here? All right, then the next investigator I have speaking is Dr. Michele Carboni, a longtime colleague and one of the major figures in all of mesothelioma, perhaps the most well-funded mesothelioma researcher at this time. Dr. Carboni is the professor of pathology and director of the University of Hawaii Cancer Center. He discovered mechanisms for asbestos carcinogenesis, implicated also SV40 as a possible role, and more recently has, in addition to the BAP1 work, been making very interesting inroads into our understanding of whether or not inflammation plays an important role in the development of mesotheliomas. It's my pleasure to introduce Dr. Carboni. Are you going to? Oh. Oh, okay. Okay. So, all right, well, moving on. All right, then, Heining, you're going to speak? Okay, then instead we will move on to Dr. Heining Yang, who is an assistant professor at the University of Hawaii, who also has been working on the BAP1 story, but has an independent program looking at the role of inflammation in mesothelioma and this role of HMGB1. Heining Yang.
Okay, good morning. Thank you very much for giving me this opportunity to share some of our research findings. Today I'm going to talk mainly on HMGB1. Later, Dr. Carboni is going to talk about more on BAP1 and probably the relationship on HMGB1 and BAP1. What we found recently is that HMGB1 really plays an important role in mesothelioma pathogenesis. So you heard earlier in this meeting by various people, especially Mary, that malignant mesothelioma really causes a significant amount of deaths. And in the United States, about 3,000 deaths per year, and worldwide there are about 100,000 deaths per year. And we all know that asbestos and aeronite exposure is one of the main causes that leads to mesothelioma development. There is a long latency, we all know, somehow between first exposure to the disease development. And there is a big variation between 20 years to 50 years, sometimes even longer. So this latency gives us a window. If there is a way we can prevent the development, so we can save a lot of life. And currently, unfortunately, only very low percentage, about 8% of mesothelioma patients were diagnosed in the early stage, stage 1, which normally has better survival. But since the patient, when they develop symptoms, is already late stage, so the prognosis is not very good, and the median survival time is only about 1 year. So there is urgent need. We all know that we need to find some better treatment to treat the patient and find the cure, hopefully soon. And here is basically an image of asbestos fibers that cause mesothelioma. So when I started my research about more than 10 years ago, I tried to understand the mechanism how asbestos cause mesothelioma. So here is just one slide showed some of the interesting puzzle, the question that confused me and many scientists in the very beginning. So we all know that asbestos causes mesothelioma. But however, in tissue culture, if you treat the cells, expose the cells to asbestos fiber, you don't see the cell transformation or become a malignant. Instead, you see cell death. So here on the left, there is normal cultured mesothelial cells. And once you expose the cells to asbestos, very few cells alive, and very soon all of them die. So how come a cell, asbestos fiber that cause cell death can induce tumor? So that, in fact, is the research question I started. So during all these years study, we found some important mechanisms that is related to asbestos carcinogenesis. And we published the paper in PNAS 2006 and 2010. And here basically is a figure summarize our research findings. We found that although asbestos causes mesothelial cell death, but the cell type, or the type of cell death is called necrotic, programmed necrotic cell death. So a protein called HMG1 is released. This is a very important factor induce pro-inflammatory response and cause macrophages and all the immune cells accumulate and secrete cytokine like TNF-alpha. So this cytokine binds to receptor, activates some cell signaling pathway, especially NF-kappa B, which makes the cell survive. But the cell survive carrying the damage caused by asbestos. So you have pool of cells which survive but has a damage. Basically, that pool of cells sooner or later will become malignant and probably leads to the development of cancer. So here I just show slides summarizing what is HMGB1. HMGB1 is a very unique protein. So in normal cells, it stays in the nucleus. However, under certain circumstances, it goes to the cytoplasm of the cells or even released outside of the cells. Only after 2000, people found that once HMG1 is released outside, it has totally different function compared to the HMG1 inside of the cells. And I'm going to mainly focus on this type of HMGB1. In fact, this extracellular HMGB1 induces inflammation that leads to the development of cancer. And also it can drive cell migration and proliferation, which leads to the progression of the tumor, which I'm going to talk later. So I don't have the time to show the detail of the research finding, which I will in fact talk more in tomorrow's science session. So I just show briefly some of the slides, which we found that HMGB1 is released outside the cells once you expose the cells to asbestos. You can see that in untreated cells, HMGB1 is in the nucleus, but in asbestos exposed cells, you went to extracellular space and also in the cytoplasm. And the release of HMGB1 induces TNF-alpha secretion, and by blocking HMGB1, you can reduce the inflammatory response induced by HMGB1. And this result we confirmed in the animal. Basically, if you inject mice with asbestos, you start seeing chronic inflammation, and if you do staining, you see a lot of HMGB1 and TNF-alpha secretion. In those sites, there is a lot of asbestos fibers. So, in fact, this result in the lab make us to think, how about human beings? How about those people who has been exposed to asbestos, whether HMGB1 also go up in their blood? So with the help of Dr. Harvey Pass, we tested some serum from those asbestos workers, as Harvey mentioned previously in his presentation. Compare those serum to unexposed people and also to the heavy smokers who have lung inflammation. And we found that only in asbestos-exposed people, HMGB1 levels in the blood is significantly induced. Compared to smoker and non-exposed, the level is low. So this makes us all think, okay, HMGB1 probably can be a biomarker for exposure, as Harvey mentioned previously. So as I showed previously, HMGB1 is really a critical factor driving the process of inflammation and also induce the tumor development. This also makes us think if we can find some inhibitors to target HMGB1 or target the inflammatory response, we can probably find the way to prevent the tumor development. So we are going to do some clinical trial that I'm going to talk later. So here is, in fact, some in vitro study which show that, in fact, indeed, by inhibiting HMGB1, we can inhibit cell transformation, inhibit the cell become malignant. Here is just one slide. I summarize that experiment. Basically, we co-culture macrophages and mesothelioma cells, expose the cells to asbestos. After about 2 months, you start seeing foci formation, which indicate the cells start get transformed. But if we treat the cells with inhibitor of HMGB1 to block the pathway induced by HMGB1, either using the boxy, which is antagonist inhibiting HMGB1, or the monoclonal antibody also targeting HMGB1, we can always inhibit foci formation. which indicate this probably is a potential novel way to prevent mesothelioma development. How about established tumor? About those people who already have mesothelioma, whether HMG1 is still critical for the tumor progression. We found that, in fact, in the tumor tissue we got from the patient, HMGB1 level is very, very high compared to normal pleural, which you can see the level is very low and HMG1 only stay in the nucleus. Instead, in the tumor tissue, HMGB1 is not just high, but also beside nucleus, is also in the cytoplasm of the cells. And it even releases to the blood. So here is a serum from a mesothelioma patient we compared with the normal people. Harvey showed these slides before. In mesothelioma patients, the serum level of HMGB1 is also significantly higher than the normal people. However, whether this level is higher or no significant difference compared to exposed people, we need to do further study to see whether HMG1 is just a biomarker for exposure or it can also be a biomarker for tumor development. So we did also a lot of study using cell lines in vitro in cell culture. We noticed that all the mesothelioma tumor cell lines express very high level of HMG1 and also its receptor rich. And we confirm this result using the staining. You can see all the tumor lines express very high, but normal cells don't express much HMG1. And the tumor cells also secrete HMG1 into the extracellular space. So that indicates that HMG1 is really critical in the microenvironment to help the tumor growth. And we did a lot of experiments confirming those data and the result. So I only have the time to show a few slides. Basically, we show that HMGB1 play a critical role, helps the tumor growth. And if we targeting HMGB1, here we use 3 different type of inhibitors, including BOX-A or the antibody targeting HMGB1, or the antibody targeting HMGB1 receptor. All of these basically inhibit the function of HMGB1. By targeting HMGB1, we can always slow down the tumor, kill the tumor cells, and slow down the tumor growth. And here is a colony formation in soft agar assay. You can see also untreated cells form big colony. So is IgG control. But if we use the inhibitor of HMGB1, including BOX-A or anti-HMGB1 antibody or anti-RIG, which is a receptor antibody, we can always reduce the size and also the number of the colony in the cell culture. Here is an animal experiment we did. You can see that without any treatment, the tumor grows pretty fast in the mouse. But if we inhibit HMGB1, here we use antibody, you can see the growth rate of the tumor is slowed down significantly compared to untreated cells. Here is another inhibitor, is a BOX-A I mentioned, is antagonist of HMGB1, also targeting HMGB1 function. This is a significant difference. This is mice treated with BOX-A, compare untreated. You can see the tumor growth is reduced, inhibited significantly. And this is mice survival with the treatment with BOX-A compared to untreated mice. You can see also the survival is very much improved. In fact, one of the mice, in the end, we couldn't even detect the tumor, so which really amazing. So we are going to conduct some clinical trial with the help of some company going to test this. I'm going to also talk later. Basically, those data I presented were published in Cancer Research 2012, and the editor even put our story on the cover page in that issue. So to summarize, basically we found that especially exposed individual and mesothelioma patients have very high serum levels of HMGB1. And HMGB1 is critical for mesothelial cells transformation and mesothelioma development. Mesothelioma cells are addicted to HMGB1, and HMGB1 is critical for mesothelioma growth and progress. So therefore, because asbestos exposed individual and the patient has a high level of HMGB1. So we think that HMGB1 can be a potential biomarker for exposure and probably can be a biomarker for mesothelioma early detection. So at this moment, we don't know. That is why we want to conduct clinical trial in Mount Sinai, which I'm going to talk in the next slides, to test whether it's good biomarker. And also, since we found that HMGB1 is critical to mesothelioma development and the cell transformation, so we think it can be a potential target for mesothelioma prevention in those high-risk cohorts who have been exposed to asbestos before and who are in the risk of developing mesothelioma. And last, we also think that since mesothelial cells need HMGB1 to grow and to progress, so we think it can be a potential target for mesothelioma therapy. So here, I will just spend some time to explain some of the clinical trials we are going to conduct very soon. Hopefully, we'll start in April this year. And Dr. Andy Todd is helping us to pull this clinical trial together. So first clinical trial we are going to do is to test whether HMGB1 can be a potential biomarker for asbestos exposure and for mesothelioma early detection. So this is clinical trial going to conduct in Mount Sinai cohort. Basically, we are going to enroll 200 asbestos exposed individuals, either insulator or sniffator, from the Mount Sinai cohort, and we will follow them for 5 years. And the CT scan at the enrollment will be evaluated for lung fibrosis, pleural thickening, and or scarring, which are the markers for asbestos exposure. And we will also have 200 age and sex matched non-exposed individuals to set up the baseline for HMGB1 serum level in the healthy population, so we can compare these two populations. And we will follow those people and measure HMGB1 in the blood, in the serum, every 6 months for 5 years. So after this study, we will be able to validate whether HMGB1 can be a marker of exposure, but also investigate the association between serum or even plasma HMGB1, the trend and mesothelioma development. So another trial that we hope to start soon is to test whether HMGB1 can be a potential target for therapy. As I showed previously that we found using inhibitor of HMGB1 such as BOX-A, we can definitely slow down the tumor growth and increase the survival significantly. So we want to test whether we can put BOX-A to clinical trial. We are working on some investigators and collaborators now. Besides that, another reagent that is already in clinical trial but in different disease called CT637 is a derivative of BOX-A, which seems to be more stable. So we are going to work with the company Criblis for conducting some study to test what is the effect and whether it's better or similar to BOX-A. Another study, in fact, I would like to mention is about aspirin. So as you heard previously in my talk, chronic inflammation is very much related to mesothelioma development. So this also makes us think, if we use some anti-inflammatory drug, whether we can inhibit HMGB1 and also inhibit tumor development. One of the well-known anti-inflammatory drug is aspirin. And aspirin is well known that it has very low toxicity, many people taking that. And also it has proven efficacy in reducing the incidence of colorectal cancer. And there are a lot of studies on that. And there are some more studies on breast cancer as well. And through my connection with Harvard University, one of my friends, she works there, and she has access to a previous study called Physician Health Study. And in that study, there were about 22,000 physicians were enrolled and followed for 24 years. And those physicians, some of them taking placebo, some of them taking aspirin, 325 milligrams every other day. So in that study, my friend take a look, and she did analysis, and she found that, in fact, there are about 17 cases of mesothelioma reported in those cohorts in that study. And after analysis, she found that there is an intention to treat analysis comparing aspirin to placebo revealed a relative risk of 0.7. What this means? This means that in the aspirin-taking group, seems the risk of mesothelioma development is reduced 30%. However, because the number is too small, after analysis, she said the number is too small to give any statistical power for the significance. However, although we cannot see there is a significant difference, but the result indicates a possible association between aspirin use and reduced mesothelioma incidence. So we did some study in the lab. So we treat the mice. We first gave the mice aspirin and some of the asbestos, and some of the mice taking or giving aspirin or another inhibitor of HMGB1. We found that the mice treated with aspirin, the level of HMGB1 in the blood is reduced significantly. And here is the mice inject with mesothelioma tumor cells. Without aspirin, HMGB1 level is high. But if we give aspirin, the level is reduced. Here is another study using aspirin. We test the tumor growth in the mice. You can see that without treatment, the tumor grows pretty fast. Here we gave the mice the garage feeding of aspirin. You can see seems the tumor growth is slowing down. Here we use another way, put the aspirin into the food, into the feed to feed the mice. And you can see also the mice eating aspirin-containing feed seems to have slower growth in the tumor. So this makes us also think probably we should conduct another clinical trial, and also thanks to Dr. Andy Todd is helping us put this together to do a clinical trial to test whether HMG1 can be a potential target for mesothelioma prevention in high-risk cohorts. So we are going to test also aspirin, whether aspirin can reduce HMGB1 level, which can tell us probably that will influence some of the low HMGB1 readings in the patient or in the exposed cohorts. But the result also can indicate whether aspirin can be a potential preventive drug to prevent tumor development in those cohorts. So this clinical trial is a prospective, double-blind, double crossover, and the dose escalation study. We will increase the amount of aspirin from baby aspirin 81 milligram up to 650 milligram to see whether that can influence the level of HMGB1 in the blood of those individuals. And for this study, we are going to enroll 40 asbestos-exposed individuals with high serum levels of HMGB1. And the participant will be randomized to take aspirin or placebo, and we will draw 10 milliliter blood to start of the aspirin, and then every 2 weeks for a total about 8 times during the study. So basically that is what we are going to do in the clinical trial to understand better what is the role of HMGB1 and what can we do to help the patient in the future. And here is the research findings that supported my study. I got R1 about 2 years ago, and recently Dr. Carbone and I, we got a V Foundation. He's a clinical PI, I'm the basic science PI. And about 1 month ago, I very luckily also got a DOD award. My research also helped by Rivera Foundation grant and also by Mesothelioma Applied Research Foundation, who in fact is one of the first award I got, which helped me to initiate all the studies. And also Hawaii Community Foundation gave me help. Thank you very much. This is a teamwork. I got tremendous support from various people, especially Dr. Carbone. And Sandra Job is a postdoc working with me, who basically developed most of the study result. Thank you very much.
ask the question? Okay. So has anyone looked to see if long-term mesothelioma survivors have lower levels of HMG B1?
Yeah, we haven't. In fact, we only test a very small number of cells, about 20 to 30 serum. So we would like to test the different survival people to see whether this level of HMGB1. We tested, although the stage, only in the tissue, we did immunostaining. We did find that the higher stage, like stage 3 and 4, have high level expression HMG1 in the tissue compared to the stage 1 to the early stage. So that indicate probably is related to survival, although we haven't tested the serum yet. Yeah.
Okay, so then I can make a suggestion from the foundation possibly that I can introduce you to the long-term survivors that are in all our groups, and perhaps we can get a study underway. What do you think?
Yeah, that would be wonderful. Thank
Thank you. Good.
Any more questions?
Laura Ferris, I'm going to have Laura speak next, then Jill O'Hara, and we're going to end with Dr. Carboni. Laura Ferris is the Director of Clinical Trials and an Assistant Professor of Dermatology at the University of Pittsburgh School of Medicine. It's delightful to have you give a presentation to us today.
Okay, thank you very much for having me here. And I am a dermatologist, so probably the last physician you thought would be up here in front of you, in addition to an ophthalmologist. But you've heard a lot of mention about skin lesions in patients with mesothelioma, and particularly people with BAP1 mutations. So I'm going to talk a little bit about that. So as we've heard about, this is sort of the initial paper with Dr. Testa and Dr. Carboni showing that germline or hereditary mutations in BAP1 predisposes individuals to mesothelioma. And here's one of the graphics that you've seen previously of two families with hereditary mesothelioma. And as you can see, the people in the orange and yellow are people who carry BAP1 mutations. Now, when we look at these families, the other things that we see are that many of them have uveal melanomas, and that's really been elucidated by the work of Dr. Harbour, who you just heard from, and the importance of BAP1 in uveal melanomas. Well, for me as a dermatologist, where this gets really interesting is that there were then reports of families who had BAP1 mutations who developed these sort of unusual unusual skin lesions. So some of these ranged from melanoma down to what look like very benign lesions, these little pink bumps that actually look fairly atypical under the microscope. Well, it turns out that some of the people who have these moles also have uveal and cutaneous, as well as cutaneous melanoma. And one group, the first group that reported on this, said it turns out this is associated with a hereditary or germline BAP1 mutation. So you saw this picture before. So as you can see, these are not really unusual looking lesions, but to anybody in the room who's looked at pathology, we know that these types of cells that have very big irregular nuclei are usually ones that are more concerning for malignancy. So when we look at the families that these lesions came from, we can see that there are many members of these families who had not only these unusual skin lesions, but also some with cutaneous melanoma. We also see people who had uveal melanoma. Now, as initially mentioned, these families, this initial group that was reported did not have mesothelioma until after this paper was published, when this patient was actually diagnosed with peritoneal mesothelioma. So now we have this connection of mesothelioma, uveal melanoma, cutaneous or skin melanoma, and these unusual tumors. And we're starting to really see a syndrome unfold. So this is where we got involved. We looked at these two cohorts that you've heard about, the Wisconsin and Louisiana families, and we knew that these families had certainly a history of mesothelioma, but we also knew that one of these families had a history of uveal melanoma, and we knew that both carried BAP1 mutations. So we wanted to see, was this a unique finding from this group over in Germany, or did we actually have a syndrome that we could identify in patients that we knew in the United States had BAP1? So we went over and I examined these people. We looked for these tumors, and these are the kind of things that I found. And I have to say, when I first saw them, I wasn't all that impressed. I thought I see things like this on patients every day, but let's take them off and let's take a look. So when we look at these under the microscope, what you'll see is that these lesions are very similar to the ones that were reported in previous papers. And when we look at these at BAP1 staining on these patients, we can see that up here and up here, we see BAP1, that's that brown stain that's in the center of the cell or the nucleus, that these are positive. And those are normal nevi or moles that were found on these patients. Now, in association with these were these more atypical proliferations with bigger cells, as you can see here, and these ones actually have lost their BAP1 expression, so showing that BAP1 seems to really be playing an important role in the development of these tumors. When we go back and we look at these patients or these families that have a history of BAP1 mutation and a reported history of melanoma, it turns out that their melanomas aren't the ones that as a dermatologist I see mostly, on a day-to-day basis. But they're an unusual variant that are called an epithelioid melanoma. And so I think you guys remember hearing that term epithelioid, which are these rounder cells, which explains some of the uveal melanoma type cells. This is from Dr. Harber's work, and you can see that if you have cells that don't have the BAP1 expression knocked out, that they're sort of stellate or angular in appearance. And then when you knock down the expression of BAP1, what you see is that these cells round up. So there really does seem to be an association with this epithelioid appearance. We see it in uveal melanomas, and it seems to be what we're seeing in the moles on these patients as well. Now, if you guys go back and read about this, a few groups have now published on this, and you're going to see many different names. So just to clarify some of this, we're actually really all talking about the same thing. So we call our tumors MBATs, and these are melanocytic BAP1-associated intraepidermal tumors. And I'll talk to you for a second in a minute why we picked that name. The original people who described these called them ASTs, or atypical spitzoid tumors. And the reason that we decided not to go with this is that as a dermatologist, the term atypical Spitz tumor is something that is a lesion that can metastasize or behave funny. Spitz tumors are normally found in kids. When they're found in adults, they're sort of a bad deal. They can have very variable appearance. And I can tell you that a lot of dermatologists will see one of these and they want to go in and excise it and treat it more aggressively. And really, in examining the patients that I examined and talking to them about their lesions, these were not lesions that were behaving aggressively. These were not lesions that were growing rapidly. And so we wanted to sort of avoid causing a lot of hysteria about these lesions. The other term that's been used are NEMPs, nevoid melanoma-like melanocytic proliferations. Again, the word melanoma in there I think would get a lot of people concerned about the risk of these particular spots. These are examples of these lesions that we've seen. All of these are benign. Okay, so why don't we make it easy? Basically, it's because we don't want to get people panicked. We don't want the term atypical Spitz tumor and have people get things like lymph node biopsies, wide surgical excisions, and the vast majority of these really behave in a benign way. And when we compare things that we look at for Spitz tumors versus atypical Spitz versus these MBATs, as we like to call them, we can see some differences. Specifically, These tumors, the M-bates, are epithelioid in shape. Spits and atypical spits also have some epithelioid cells, but they also tend to have a noticeable spindle cell, or those kind of more long cells component to them. The ones that we see, the M-bates, are really deeper in the skin, in the dermis. Most spits and atypical spits tumors are up higher in the skin, in the epidermis as well as the dermis. In terms of mitoses, or division of cells, one way to measure this is there's a stain called Ki-67. We found that these tumors had lower levels of Ki-67, suggesting that they're not rapidly growing and dividing. We also found that most of ours were in association with a normal run-of-the-mill nevus or mole on the patient, and that's not the case for some of the others. So there are some other things that we do see that I'll talk about in a little bit that may suggest that some of these lesions could progress to more aggressive ones, but most of what we saw in the histories we got were that these were benignly behaving. So what's the difference between these things and a melanoma? Here are these M-bates, and these are pictures of melanoma. So we're really kind of talking about two different types of tumor. Certainly, these things are bad. These are the things that need to be treated rapidly and identified, but these are very different in appearance. Now, can these things ever turn into melanoma? We have some anecdotal evidence in the fact that some of these patients do have a history of melanoma when you look at their family trees or their pedigrees. But here's an example of one melanoma that was removed from a patient who carried a BAP1 mutation. And what you can see is at the base here, this is one of these epithelioid lesions. The nuclei don't look, too awful in here. They're smaller. However, above it is this big, expansive proliferation that isn't one of these M-bates, but is actually a melanoma. We could see really atypical dividing cells like that. So this would suggest that perhaps this melanoma evolved from it, but not that, from this little more benign lesion. The other thing that we found is that we did look for this gene BRAF, a mutation in BRAF, and BRAF mutations are found in some melanomas. They're also found in some Spitz nevi. However, it does suggest that perhaps, as a previous speaker mentioned, that when you have one gene and then you start to accumulate several other genetic mutations, there may be a risk of some of these progressing to melanoma. So one group did actually look at sporadic melanomas, not ones that were from mesothelioma patients, and they found that 5% of melanomas will carry the somatic, so the acquired later BAP1 mutation. So we think that BAP1 can play some role in melanoma, but it's probably not something that is a major player in cutaneous or skin melanoma the way that it is in the uveal or the eye melanoma. Another way of looking at this is that one group that follows patients with hereditary cutaneous or skin melanoma, they took a sample of 200 families or individuals from families with hereditary melanoma, and they divided that into which people have both skin and eye or uveal and cutaneous melanoma, and they found that there's 7 families that had a history of both, and then they found that 193 families had a history of skin-only melanoma. So it turns out that of those 7 families with skin and eye melanoma, 2 families had germline BAP1 mutations, or 28% of those. So a fairly high rate if you see both of those in one family. However, if you look at the people who don't have a history of ocular melanoma and only have skin melanoma, they only had 1 family, or less than 1%, that carried that. So that suggests that the hereditary mutation of BAP1 is more strongly associated with the family skin plus eye melanoma, rather than with the hereditary skin alone. When we looked at, in our paper where we looked at the families in the United States that had these BAP1 mutations, and then also looked at the other reported families with BAP1 mutations, if you do some analysis, you can see that having, there's about a 17-fold increase in risk of having at least one cancer if you carry a BAP1 mutation. So this really goes along with the idea of this BAP1 cancer syndrome. Certainly if you look at mesothelioma mesothelioma. The only cases of mesothelioma were among patients with the BAP1 mutation, as well as uveal melanoma. Interestingly, we also see that with if we try to get histories of cutaneous or skin melanoma, and that does seem to be statistically significant. And as well, the M-BATEs, or these tumors, really were only seen in people who had known BAP1 mutations. For other cancers that have been suggested, we didn't see it, but again, you need large numbers to see less common tumors. So really, we're looking at, is this a BAP1 cancer syndrome? And we do know that certainly uveal melanoma, certainly mesothelioma, and it would seem cutaneous melanoma are things that we may be at risk at in people who have BAP1 mutations, as well as these M-BATEs, or these tumors. Other tumors that may be associated include breast, renal or kidney tumors, lung cancer, other ones including bone marrow or blood-borne tumors. One group actually coined a term for this, the common syndrome, which is cutaneous and ocular melanoma, characteristic melanocytic proliferations, and other internal neoplasms. So what, as a dermatologist, or what can we take away from this? So what we've found when we study these M-BATEs is that they tend to develop during the second decade of life, so earlier than people would be diagnosed with other cancers in general, like uveal melanoma or like mesothelioma. And I think that the role as dermatologists, what role can we play? We can help to identify these people. So it's not that everybody who has a pink bump, we say, boy, you may be getting mesothelioma or uveal melanoma, but it's that when we do get these things that look pretty benign clinically, but then we biopsy them and we get back this report of an atypical Spitz tumor or an epithelioid tumor, we probably need to go digging a little deeper. We need to ask, do these anyone in your family have a uveal melanoma? Has anyone in your family had a mesothelioma? And we can maybe help to identify people who may be at risk for these tumors early, because this is such an early marker. And so, as we know, there are things that we can do to maybe help lower the risk of some other tumors, whether it's strict avoidance of asbestos. Certainly we could counsel people in terms of what kind of career they may choose or what kind of housing they may choose to live in. And as a dermatologist, we're always looking for ways to encourage people to use good sun protection as well. So, mesothelioma is bad enough. Do I have to worry about melanoma too? So for most people with mesothelioma, the risk is not going to be higher than in the general population. For people who have BAP1 mutations, there may be a slightly higher risk. And so the things that you can do are protect your skin and eyes from the sun. And I can't say that's only for people with BAP1 mutation. I mean, that's for everybody. Sun protection is key to preventing melanoma. Most melanomas are not going to occur in people who are genetically at risk. They're going to occur in the general population. But certainly if you know that you have a higher risk, remember sunblock, remember wearing protective clothing, and remember wearing things like sunglasses. Okay, so I have this pink bump on my skin, so that's really bad, right? No. These are lots of pink bumps that I see every day on patients. If I looked at everybody, I'd find a pink bump of some sort. Every pink bump is not an indicator of melanoma or of a risk of melanoma or mesothelioma. All of these are harmless and common. So if you do carry a BAP1 mutation, it's probably reasonable to see a dermatologist starting at the age of 18 annually for screening. And in terms of ophthalmology, I don't know what kind of screening they do, but certainly talking to your ophthalmologist about the risk. So most of these lesions are not going to, these M-BATEs or whatever you want to call them, are not going to progress to melanoma. We would never recommend go out and get every single pink bump on your skin, removed and excised at this point. It's just like most of us have moles, little brown nevi or moles on our skin. Nobody would ever recommend that you remove all of them, even though maybe about half of all melanomas start in nevi. It doesn't mean that most of them are going in that direction, are going to to progress to melanoma, we look at it as a marker of the more moles you have in general, the higher your melanoma risk. If you have M-Bates, it's a marker that perhaps we need to follow you more carefully or look for a germline BAP1 mutation. If you're going to have a dermatologist follow you, make sure that they understand what this gene is and what this does. I can tell you, when I presented all this work to my colleagues and my residents, none of them had ever heard of BAP1. So most dermatologists aren't going to know about it. You may need to be the one that says, hey, what about this gene, BAP1, and if you find something, I want you to know that I have this. The other thing is to make sure that whoever, if you do get a skin biopsy, the pathologist who's reading it is aware of this as well. So one, you'd want a specialized skin pathologist, and two, you'd want to make sure that they've heard of these. So you don't want them to read this as an atypical Spitz tumor and have the pathologist who may not understand the syndrome and the dermatologist who may not going in and recommending really aggressive care. So that's it. Thank you very much.
The next speaker is Jill O'Hara. Dr. O'Hara is professor of medicine in the section of pulmonary, critical care, allergy, and immunologic disease at Wake Forest University in North Carolina. It's a pleasure to have Jill speak to us.
Well, thanks a lot. I'm very honored to have a chance to speak to you today. This is an update of research in progress. I guess I should kind of start out, though, to say that I've learned a lot already this morning, and this has been a great symposium. Mostly I learned from Dr. Harbour about stem cells. I really never thought of them this way. And that is that they kind of reminded me of my ex-husband. They tended to revert back. They were uncontrolled and exhibited dangerous behavior. So, again, okay. So, you've seen a lot of these slides before. Have you noticed we're all using the same slides? But that will allow me to go a little more quickly because of time constraints. It's already been mentioned that somewhere around 30 million Americans between 1940 and 1980 were significantly exposed to asbestos in their workplace. This does not in any way, this large number, in any way reflect all of those bystander exposures, the wives who washed the clothes, the kids who crawled up on daddy's knee after he came home from work with his dirty overalls on. Despite that very vast number, there are only about 3,000 new cases of mesothelioma diagnosed annually. It's estimated there's probably an equal number who die and are never diagnosed, but still 6,000 a year versus 27 or 30 million is a startling gap. This is my attempt to kind of make some sense, some rationality of that. We've heard a lot about genetics today. Certainly, we've also heard a little bit about inflammation and immunology. It seems that age at first exposure may be important. We published a paper that showed that the younger you were when you were first exposed, the better the chance that you might get mesothelioma. There's a precedent in that in both lung cancer and COPD, with the earlier you start smoking cigarettes, the better the chance you'll get those, even when you factor in the total number of cigarettes that you've smoked. Gender, are women more prone to mesothelioma? There are some older papers that would seem to imply that, and clearly the longer the latency, the better the chance you'll show up with a mesothelioma. And then finally, dose. And you can see there are people who have huge doses that have a pretty good chance of getting mesothelioma, and very little in the way of a genetic predisposition. Equally, there may be people down here who have a huge genetic predisposition and maybe little or no asbestos exposure whatsoever. This is just a hypothesis. Now, I'm going to update you on what we're doing to try to look at this. We believe that there probably is a genetic predisposition, and so several years ago, I started collecting blood to isolate DNA on people who had mesotheliomas, with the idea that we would do a genome-wide scan. Since that time, we've become enamored with the BAP1 story, and so I'll tell you a little bit about what we're doing with that. But to date, you can see that we have 441 samples. We started a consortium to share the DNA, and that was funded by MARF, and you can see the centers that have contributed to that thus far, and we are still accepting samples. We also noted in our large cohort that there was this very strong cancer signal. When we looked at the first-degree relatives of people who had mesothelioma, we saw that overall there was about a threefold greater chance that they would have a first-degree relative that had a cancer. And if we looked at their kids, and remember that cancer's just generic cancer, colon, breast, prostate, lymphoma, those kinds of things, these are usually age-dependent tumors. That means the older you are, the better the chance that you're going to come up with a cancer. And when we looked at these mesothelioma victims' children, they had about a 7-fold greater chance of having a kid with cancer compared to our asbestos-exposed controls. Now, there have been a lot of tumors that you've heard about today that may have a link to BAP1 germline mutations. So what we did is we looked at our population, so overall 441 mesotheliomas, and we wanted to look at just those who had a very strong personal and family history of cancer. Furthermore, we have a control group of approximately 3,500 asbestos-exposed individuals who have asbestos-related disease in the form of pleural plaques or some fibrosis, but do not have a cancer, or do not have an asbestos-related cancer. And we looked in that group of 3,500 to look for people who had no personal history of cancer, no family history of cancer, and who had the longest latency. That means the time from they were first exposed to asbestos until we evaluated them, so we could give them plenty of time to develop a mesothelioma if they were going to. We then created a cancer epidemiology scoring system. And so if you had one condition, like a personal history of having had a cancer before you got your mesothelioma, you got a point. If you had a parent that had a cancer, you got a point. If you had a sibling, a brother or sister who had a cancer, you got a point. If you had a child with a cancer, you got a point. Furthermore, we thought we'd seen in our families that sometimes there were a large number of brothers and sisters who had cancer. So we gave you an extra point if more than half of your brothers and sisters had a cancer. We also gave you an extra point if you had a kid with cancer, because as we mentioned, this age relationship with the development of cancers overall. And finally, you got an extra point if you had a personal history or a family history of having a cancer that was potentially related to BAP1. So these are the centers then that contributed to the BAP1 cohort. In all, there was 151. And we also came up with 154 controls. And as I mentioned, we wanted to look for people who had a very long latency in our controls. And you'll note that many of the people that we have in our cohort are workers and therefore blue-collar workers. And today, and even in the old days, if you wanted to find smokers, you didn't look at doctors and lawyers and Indian chiefs, but blue-collar workers even today tend to smoke more frequently than do librarians, who have the lowest prevalence of cigarette smoking in the United States. Are there any librarians here? Good, let's give that person a hand. All right. But as you can see, the latency was longest for the control group. Age at first exposure, our mesos tend to be very young at their first exposure, often in high school jobs, jobs after school, in high school and in college. Our cohort is unusual because it's workers, so it has mostly men in it. When we looked at the history of cancer, you'll note there's no control group here because all of this would be zeros for them. We picked them, handpicked them, that they had no personal history of cancer in the past, no parents with cancer, no siblings with cancer, no children with cancer. And you can see that overall our mesos tend to have that high prevalence, but this group that we used in the BAP1, or that we're using in the BAP1 project, have a particularly high rate of cancers, and that's by design. You've seen this already. You've seen this slide. It's a picture of the BAP1 gene. We're sequencing this gene in all 305 of the patients that we've picked out, the 151 mesotheliomas and the 154 controls. To date, we've sequenced just 28 samples. The samples were given to the two centers, Fox Chase and our center, in a very random fashion by design. So you'll see that the randomization has ended up that only 5 cases have been sequenced to date and 23 controls. This is, of course, continuing even as we speak. As you can see, these are the known mutations that have already been presented for the uveal melanomas. Next is what we have so far. These are the mutations that we have seen, and the red ones are our cases, and the blue are our controls. No, I take that back. Our blue are our cases, our red are our controls. And what we can see at this point is that while there are several SNPs that we see, most of them are in non-coding sections. Now there are a few here at the splice site, or near the splice site, that may in the end be important, but we have no knowledge of that yet. There also are a few down at the untranslated region, the 3' region, that may also have functional relevance, but we don't know that yet. So this is not to enlighten you, but just to tell you what's ongoing at our center. We'd like to thank all of you who have been involved so far, who've given us blood. We would like more, obviously. And so if you are interested, if you have a mesothelioma and you're interested in giving blood, please contact Mary. She knows how to get a hold of me, and we can enroll you into the study. These are the numerous folks who have contributed to this study so far and the ongoing work. So thank you for this opportunity to speak to you.
Our last speaker is Dr. Carboni, and I've already introduced him. He's still the Cancer Center Director as of about an hour ago. We just lost our Cancer Center Director instantly. You know that.
Good morning. It's a pleasure to be back at MARF after many years. Several of you asked me yesterday night, how is that it was many years that I was not here. I have no idea. The time passed very fast. But in fact, it has been several years. Last time that I came here, I think I thought about what at that time some people thought was a crazy idea, that there was a gene that caused mesothelioma in certain families. In fact, I remember that there was a lawyer who listened to me at my talk, and he published an article that said that he said that Dr. Carbone, rather than wasting his time going to Cappadocia and to rural areas in America, should go to the Caribbean and enjoy life, because there is not such a thing as a gene. The newspaper guy, the guy who wrote the article, called me, wanted me to comment. I declined that. So we did a lot of progress since that time. You hear a lot about this gene today. I am not going to give you a formal presentation. I had the pleasure, the fortune, whatever you want to call it, over the years to work with, for more than 10 years now, with Dr. Testa, with Dr. Paz, with Dr. Young, sorry, and more recently with Dr. Ferris. And I've worked as a team with them, and I would inevitably repeat many of the things that you heard, and I think that you heard enough today. So I'm not going to give you a formal presentation. I'll just make a couple of comments. What does all this mean? What is this BAP1? Well, obviously it means a lot to people who carry the BAP1 mutation, but most patients with mesothelioma do not carry a germline mutation. Still about at least a third, but probably more than that, probably half of them carry BAP1 mutations in their tumor. So what that tells us is that this BAP1 gene is very important in the process that causes mesothelioma. And so now we are studying how this process goes, what happens, we really do not know how this BAP1 works. We have a lot of hypotheses out there, but we do not know how it works. So we are studying how we can, by understand how BAP1 works, to develop specific therapies for mesothelioma, because you know that we do not have specific therapies for mesothelioma. At the same time, we discovered that it was not just mesothelioma, but there is melanoma. Who would have thought that melanoma and mesothelioma had something in common? Nothing. And so people who are experts in the field of melanoma are coming into the mesothelioma field, but also we can learn from what has been done in the field of melanoma and see if we can apply something from the melanoma fields to the mesothelioma and vice versa, since now we find that the pathways that give rise to these tumors are the same. You heard a lot about how these genes work in the previous presentation. I have a paper that is out this week together with some of the people that I mentioned before, with Dr. Testa, with Dr. Paz, with Dr. Young, in Nature Review Cancer. The paper is out this week. I will give a copy, I forgot in my room, to Mari, so that this afternoon will be out there. If you want to know more about this BAP1, everything that is basic science will be there. And for what is the clinical part, I already gave the 50 copies that I had of the paper that we published in Journal of Translational Medicine with Laura Ferris, that she showed the slides before. And there we discuss all the clinical aspects of this cancer syndrome. So if you want to know about the clinical trials and all this part, you can take a look at this paper. The other part that we have learned is that genetics by itself is one factor, but inflammation is a critical factor. And in fact, inflammation is what drives the process of carcinogenesis and possibly the growth of these tumor cells. You heard the work of Hein Yang, and how, in fact, by targeting inflammation, we may be able to delay tumor growth. And you know, if you can delay tumor growth in a tumor that appears in the 70s and 80s, well, if you can make it appear in the 90s and in the 100s, you have done a lot. So we want to see whether we can delay tumor growth or whether we can use some of these inflammatory markers for early detection of this tumor, when, as you heard from Dr. Paz, the tumor is much more susceptible to therapy. The other part that I wanted to tell you is that this work has been possible because of teamwork. Now, teamwork in science is a difficult thing to do. And the reason is not just that scientists are naturally egoistic and full of themselves and want to compete with each other. I mean, there is certainly some truth into it. But the main reason is that the system is set up in such a way that does not favor people to work together, even if in principle it does, because we go ahead in our careers by publishing papers, by getting grants. Our institution wants the grants to come to our institutions, and therefore we are naturally competing with each other. Instead, what we have done in the mesothelioma field is to create a network of some of the top researchers in the country and internationally who have worked together, who have shared what we had and different expertise. And that's how, for example, this BAP1 gene was found, and that's how all this research has been found. And as this team enlarged, other people come to the field and bring their expertise to it. You hear today, we have here today Dr. Ferris. The reason that Dr. Ferris is here is that she's one of the top experts in melanoma, and so when I needed somebody who was an expert in melanoma, I called Laura. Now, Laura is in Pittsburgh, I am in Honolulu. Still, it doesn't make a difference, because nowadays, thanks to Federal Express, email, airplanes, in fact, with Laura, we did 2 trips in Wisconsin and in Louisiana to collect samples from these families that we are studying. In fact, one of the families I met here, the Louisiana family, was thanks to the Marr Foundation that I met them here, and that's how we were able then to follow them. MARF has been instrumental in supporting this collaboration. Marius Dorfer in particular has been, I do not know where is Mary now, but Mary has played a critical role, in fact, in the collection of samples that allowed us to identify the BAP1 gene. Without her, we probably would have done it, but later. And so she played a critical role. And she continues to play a critical role in enrolling families, because we need the largest possible number of families in order to come down with some minimal results. If we have few families, we do not know what exactly something means, as you heard from Dr. Haber. But the more we have together, the easier it is to study. So Mary is, in fact, continuing to play this role in helping us enlarge the number of families that we are studying. So if you know of anybody who suspects to have a familial condition, please have them contact Mary, because the larger the pedigrees that we have, the more quicker we can do progress going forward. So we have really done faster and better in mesothelioma because of teamwork. The Marfa Foundation has allowed us to have a forum that provided to expand this network of collaborations. And in fact, together with them now, we have submitted to the NCI one of the largest grants application that the NCI funds, that is called SPORE. There has never been a SPORE funded for mesothelioma. It looks that our application was received well. I do not know whether it's going to get funded this year or not, but it would be a fantastic thing if we can bring a SPORE, because it would mean a tremendous amount of resources to the mesothelioma field. And SPORE's grants are grants that are designed, in fact, for teamwork and to get people from different angles to study the same disease. So I'm very grateful to Marfa. I am very happy to be here again, and I'm very happy to be here now that the gene, in fact, has been found, and that half of the talks today, are actually 3/4 of them, were about this gene, which, having been here a few years ago, just talking about the hypothesis after which I was after, I have to say it was a great satisfaction to see that in fact, that has become true. Thank you very much.
Well, I think this was a terrific session, and I hope you all feel the same way. I will leave you. I understand everyone wants to get to have some lunch, and that's so I'll make my remarks in about 2 minutes. Despite what we know now, there are already some issues that are arising that are extremely interesting and somewhat alarming. For example, I have one family that contacted me that do not want to be tested after initially thinking they should be tested because they're afraid that their children will not be able to get insurance. I'm told by many of the people in our family risk assessment program that that is not the case. So I think we will have to get that message out to folks because knowledge is strength for all of you. Secondly, I think from a biological point of view, there are some very interesting questions. Why does a BAP1 mutation occur in the germline and predispose somebody to getting cancer? And yet Dr. Harber has elegantly shown it can also be a mutation that leads to metastasis. How does a gene do both of those things? We had 2 squamous cell carcinomas in Louisiana family. Are those cases really squamous cell carcinoma, or are they some other kind of skin lesion that we weren't aware of before? Not all of those individuals did we have access to them with a dermatologist at that time. Thirdly, uveal melanoma is, in all of the textbooks that I've tried to search this out on, listed as not having an effect of ultraviolet light, sunlight, on this disease. Is it possible that if you have a germline mutation, that makes you exquisitely sensitive to getting uveal melanoma? Remember, those studies that are done that say there's no connection are done in the general population, 99.9% of whom will have no germline mutation. So that's another question I think we need to address. Along the same lines, is asbestos really going to be essential? I've already been contacted by several lawyers. I don't know if there are any here, but sometimes it's very disturbing because I get the sense that some of the lawyers are calling simply to try to get their clients off from paying what they really owe the patients, which is a settlement for asbestos exposure. To my knowledge, there is no evidence. We're going to do this in mouse studies. I'm sure other groups will do it as well, to see if mice with BAP1 mutations are more sensitive, more likely, more vulnerable to get mesothelioma than a wild-type mouse. But the reality is we have really no evidence that a person who gets mesothelioma with BAP1 mutation gets it just because of the mutation. I think everyone, probably even in this room, there's asbestos. Nobody walks the walk without being exposed to asbestos. Even one paper reported by Weissner, I hope he's not here, but I know he told me personally that a family he reported that had no exposure to asbestos, he told me that family had lived in an apartment building where they removed the roof. The roofs in Europe often have asbestos in them. In removing that asbestos, there's no reason to suspect you couldn't get some of that to come into the air conditioning system and expose people. And there is no threshold for safety. You don't say, okay, I'm okay as long as I don't get this much asbestos. We don't know that there's a safe level of any asbestos. The other thing I think is, why do some people get mesothelioma and others get uveal melanoma, others get cutaneous melanoma? One of the pedigrees that was placed up here showed not one person in that family getting mesothelioma. Why is that? And what causes, and we don't even know this for P53, what causes a gamut or spectrum of diseases to be to have a particular mutation? For example, in Lee-Fermini syndrome, why these particular tumors, some of which, like adrenal cancers that are kind of uncommon, why are they getting the mutation causing that type of cancer? It's quite possible that there may be things at the expression level of different cell types that might make them common at the epigenetic level that we can't imagine based on their histology. And finally, I think Dr. Ferris gave a very good lesson for us. We want to be learned. We want to get information. Knowledge is great. We don't want to become crazy about this. I mean, I think that if a person does have a lesion, get to a very experienced dermatologist. But there's no sense getting crazy about the fact that you have some little pimple on your skin. And finally, I think for all the families who are here, like Dr. Carboni, I want to express my deepest appreciation for the support that the MARF has given. In fact, one of my colleagues at Fox Chase recently received an award to do that very kind of study, to look at 2 different mouse models where we've made mutations that are exactly like the mutations in Louisiana family and in the Wisconsin family, to see if they do have susceptibility to mesothelioma spontaneously, or if you give them asbestos, do they get a higher rate of mesotheliomas than their wild-type littermates. So I want to end with that and thank you all very much. And any of the cards that are where you have any additional questions, I think there are plenty of opportunities, Mary's indicated, during this meeting we'll be able to address those questions in another forum. So I thank you all very much for joining us today.